this is titled, this will be titled in your Canvas discussions. I did this with the morning class already, but I'll do it with you and we'll upload you, and that's what we'll do. So again, this will be loaded up during your break. This is chapter 2, chemistry, part 2. There's not going to be a part 3. Okay. All right. So let's go. First of all, am I recording? Yes, I am. All right. So we left off here in this chapter, and this is talking about carbon, how awesome carbon is. Remember again that we have valence electrons on the outermost shell, or farthest away from the carbon. Those are your valence electrons, and those are responsible for the game of chemistry. Those are the ones that bonds are taking place at. So if you look here, the hydrogen has 1, these have 4, and as a result, these are sharing electrons. So it is the valence electrons that are taking place, and this is known as the covalent bond. We will get into polar covalent, non-polar covalent, but in a little bit. So then we take a look at carbon, and carbon here is also bonding. It's also bonding to 4 things. So it's bonded here, 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 and here, and that's 4. So this is known as a carbon backbone. This is glucose. Glucose is the most important sugar of all your monosaccharides. And it says somewhere that there was a hand raised.
Yes, I did. Can you hear me?
Yes.
I had a question. So for our classes, you're not going to go over, you're just going to post a video, and that's going to be like the part 1, and then each class just goes through a different part. We're not going to go through the whole thing?
Yeah, we're going through the whole thing right now. Well, we already kind of covered part 1.
We did?
Yeah. Like we only need, we're in the middle.
When did we cover part 1? Because last week we didn't have class. It was Labor Day.
Oh, maybe you are correct, and I stand corrected then. But want to start all over again? I mean, it's posted, it's already recorded. Do you want to do it? We could do it. I have no problem.
It's up to you, Teach.
I think we should start with the halfway point, considering that, again, you're right. I forgot about, we didn't have Labor Day, and you're a once-a-week class. I don't think it would be in your best interest that we go back and reinvent the wheel. You know what I mean, Jelly Bean?
All right, perfect. Then let's keep going.
Yeah, so it's there. Have you seen it, or have you looked at the lecture already?
I saw the part 1 was posted. I haven't looked at it.
Okay, so that's good that at least you know that it's posted. That's more than some. So that's good. You're off to a very good start. So please take a look at the part 1. Sometimes, I apologize, that was my phone. Let me try to, yeah, sometimes, yes, in a situation like that, and Ms. Mendes brings up a very good point. Situation like this where we had a Labor Day, thank you for actually this very good question. Yeah, we're gonna have to go this route. And fortunately for you, I'm not telling you, oh, you know what? That's independent study. Go review it. Because guess what? You already have a lecture there for you. So there's this situation. I know we're in this COVID-19, but it actually helps students, because if we were in class, we wouldn't be recording it, because there's really not a way to record that when we were in class, unless you're recording me with your cell phone. So in a way, this is very, very good for you that we have this situation. But okay, we're going to pick up at where part 1 ended. All right, we're going to take off where part 1 ended. All right, so let me share screen. All right, so here we are. We have carbon. I already mentioned how important it is, and it bonds mainly to usually to 4 things, but sometimes it can bond to 2 things. For example, carbon dioxide, not to be confused with carbon monoxide. All right, so I'll probably be asking you that on your exam. Now, here we have oxygen. This is the oxygen we breathe in, O2. This is not singlet. So if I cover this and it's actually just a oxygen by itself, that is known as singlet oxygen. That is an ROS. That is a reactive oxygenated species. Don't look that up in your book. It doesn't exist. I doubt it. But I'm telling you. So singlet oxygen, it can be very damaging to cells and also to DNA. So singlet oxygen, not a good thing. Now, 2 oxygens like that, a very good thing. That's the oxygen that we need. Add another oxygen, O3, and now we're talking about ozone. Very, very good in terms of atmospheric ozone because it absorbs ultraviolet light, which is really good because that can hurt all life on Earth, plants, animals, cats, dogs, humans. But in the case in which ozone is in ground level, that can be pretty bad. Now, you're probably wondering, how does ozone from up there get down here? Well, it doesn't come down. What happens is that due to certain high temperatures in the summer and congestion of cars, you could have various emissions released from the combustion engine due with heat, and you can have O3 created. And that's how come some cities do a carpool lane so you can reduce the flow of traffic. So you and your buddy can go to work, you can get to work faster, and as a result, you can also help reduce emissions, which create ozone, ground level ozone. That's how come if you've ever been through certain big cities, in summers, they may say dangerous ozone levels. So in case you're wondering what agency is doing the monitoring of this ozone levels, it is TCEQ, the Texas Commission on Environmental Quality, then contacts the Texas Department of Transportation, in which then they put up those signs saying, hey, dangerous ozone levels. Individuals at risk are elderly and the young. So young children, developing lungs, and as well as elderly or any individual that has pulmonary issues, for example, asthma. So please remember singlet O2, O3. Here's this wonderful molecule that brings life to us, and that is water. What we're going to do now is we're going to jump a few slides, and we're going to talk about water. I'm going to introduce the concepts of polar, non-polar covalent bonds, and then we will jump back and we're going to take a look at macromolecules. Trust me, this is the best way to present this. So we are going to jump into water. Oh, let me escape. Don't worry, we'll come back to this. Here we are. All right, let's talk about water. We have here a picture of water. Right here. O2, oxygen, 2 hydrogens. Now, whenever you see any dashed lines or dots to another water molecule, that is the hydrogen bond. So what bonds 2 water molecules together? The hydrogen bond. What do these little dots or dashed lines, some textbooks use dashed lines, that is the hydrogen bond. The hydrogen bond is not taking place within the water molecule. What is binding the H and the O? That is called a covalent bond, okay? So here, 2 water molecules, hydrogen bond. Here inside this is your covalent bond. Now, I'm going to explain to you polar covalent. So, think of these electrons as toys, okay? Think of them as toys. And one of these electrons belongs to hydrogen, and one of these belongs to oxygen here. Okay, now, if you've ever seen children play, if you tell them to, just because you share, that doesn't mean you are sharing equally. So if we were to do a little dashed line slightly above the little O right here, we could see that oxygen has 1, 2, 3, 4, 5, 6 electrons or toys. So definitely oxygen is hoarding these toys, i.e. electrons. Well, since this kid, oxygen, is not sharing equally, we have something called a partial negative charge with the delta here. So this oxygen here has a more of a negative charge. So for example, right here, this is more of a negative charge here. Now, what happens is because of this hoarding of these toys, i.e. electrons, there is more of an electronegative pull in this direction. And as a result, there is more of a positive or partial positive charge right here in this location of the hydrogen. All right. Now, because of this unequal sharing, it results in something called a polar covalent bond. Okay. A polar covalent bond. So water is polar because the electrons are not shared equally between the oxygen and the hydrogen atoms in each of its covalent bonds. Okay. And everything that's in here, pretty much I've already explained it in my own way, but feel free to read it. So we have that situation. Now, when you have a molecule that is doing equal sharing, it is going to be called non-polar covalent. Very important concept. Very, very, very important concept, because when we go into the macromolecules, I'm going to start talking about, which is the material that we skipped. I'm going to use the word polar. I'm going to talk about the word nonpolar. All right. And if I would not have gone through this part, you would have been lost. So that is why we're jumping here. Okay. Let's talk. Okay, we already talked about many of its functions are due to its shape. Yes, shape, but mainly due to its configuration. Yes, shape would be considered the term stereochemistry. And we're going to look at, okay, we already talked about polar, unequal sharing. We got it. Partial negative, got it. We have that partial positive, got it. Electrostatic, all right, we already talked about, yes, hydrogen bonds, we got it. And then we're going to talk about these hydrogen bond gives water its unique properties. What type of properties are we talking about? Well, here are just a few. I will mention more. Cohesion. So whenever you see a drop, you see them hanging out together, these water molecules, they're kind of a little on this little sticky, clinging side. They cling together to other water molecules. That is called cohesion. When water molecules cling on a surface, like your skin, you get out of the shower and you have to dry off because the water is just not all just beating off you. That is called adhesion. Okay, adhesion. Now, water at 0 degrees will freeze, and at 100 degrees begins to boil. So I do not think, it doesn't use the term, but has a high heat capacity. Water has a high heat capacity. So for example, you get water from the tap and you put it here. Then you turn on this burner. Well, it doesn't immediately start burning. I'm sorry, not burning, boiling. It doesn't start boiling. Why? It's because it's absorbing that thermal energy until it reaches the point of 100 degrees Celsius and water begins to boil. And then water can change into another physical state. So water has 3 physical states. It is a solid in terms of ice. It is a liquid here. And then it can turn into a gas. Very, very unique. Not many molecules can have multiple physical states like a liquid, a solid, and a gas. This concept here that I'm introducing to you, a high heat capacity, is one of the reasons why we have such fluctuation in temperature here in this area. So we have, when we start approaching winter, we have these drastic temperature changes that can occur from anywhere between 40 degrees and 45 degrees. That is a heck of a change, degrees Fahrenheit. So in the day, it can be hot, and then at night is cold. So what in the world is the reason for this massive, drastic change? 40 to 45 degrees Fahrenheit change within 24 hours. That's a little ridiculous, but it does happen in this area. That is because of the lack of large bodies of water. Water, as I've explained in this situation where you see that pan and the burners and boiling, it's absorbing heat. Well, the same thing with a large body of water. And when I mean large body of water, I'm not talking about a little pond. It has to be a good sized lake or an ocean. And it absorbs it absorbs heat. And then during cold times, it can release that heat and there's like a balance. That is how come areas that have large bodies of water, their temperature fluctuations do not drastically change like ours, because water is what is maintaining that temperature. We don't have that, okay? So because of that high heat capacity. Now, let's talk a little bit about density. Density is the amount of matter per volume, and due to the hydrogen bonds, when water freezes, it forms this lattice structure in which creates, it has less density than other solids, and as a result, it floats on water, right? Now, make no mistake, ice, it can be heavy. Ice can, if you put ice, cubes of ice in a sock and you whack someone over the head, like it's gonna hurt. Who knows? You could potentially, damage somebody by doing that. So just because it's water, that doesn't mean that it can't hurt. Now, with that said, look how water can give life. Water can also take life. For example, you look at a tsunami or a flash flood, those can knock down buildings, it can break bridges, and it can take life. So water has to be respected. Now, there's a lot of energy, lots of energy within those hydrogen bonds, lots of energy. So individually, that hydrogen bond is very weak, but you add a bunch of them together, and they can be very, very strong. Any questions in terms of water, characteristics, bonding, polar, nonpolar? Okay. We will continue in this direction, and then we will go back. Trust me, this is the best route. So a solvent, this is what is doing the dissolving. Solute is what is being dissolved, the substance being dissolved. And a solution is when you have solvent and solute. Okay, I'll give you some examples. Water is a very good solvent. It is a common solvent. And that's not to say that there aren't others. Now, if you have Kool-Aid and you put it in a glass and you put water and you stir it, the water is the solvent and the Kool-Aid is solute. If you're making some sort of tea, the tea leaves will be the solute and water will be the solvent. You're making coffee, and yes, the water is heated, but that's to extract the components, for example, caffeic acid from the coffee grinds, and the water is the solvent and the coffee is the solute. Okay, let's take a look at this now. Let's talk about things called ionic. Now, ionic means charged. It's going to be charged, an atom that is charged. So here we have sodium, and it is positively charged because there are more protons than electrons. So if you have more protons, protons are positive, electrons are negative. Even if you have one more, it's going to be one more proton than electrons, total electrons, by the way, it will be positive. In the case of chlorine, if you have more electrons than protons, remember protons in the nucleus, it will be negative. Well, it just so happens that opposites do attract, and sodium, positive, and chlorine come together to have sodium chloride, also known as your table salt. And what we're going to do is we're going to discuss a little bit of hydrophilic, hydrophobic interactions a little bit here. We're going to talk about the attack. So let's say you are going to add some water into a warm glass of, you're going to add salt to a warm glass of water because you're going to gargle, you have some allergies, maybe you have a little bit of drainage in the back of your throat, a slight little film of mucus, you want to clean that throat. So you gargle with warm water and salt, right? And you stir it. So you're going to dissolve this. This is the crystallized structure of salt. All right, well, how does it dissolve? Well, what happens here is if we take a look at this chlorine, right? This is the chlorine, and it's negative. The hydrogens, this is water inside, water, the hydrogens here are partial positive. Then the blue parts of water or the oxygen, that's partial negative. So you have this negative chlorine, and then these water molecules are gonna come up to it and be like, yo, yo, what's up? What's up, chlorine? What you gonna do about it? And the water molecules are going to face with the hydrogens toward it, and they're going to attack it and surround it and kind of like jump it and just surround it and stabilize it, okay? The same thing happens with sodium, which is sodium's positive. So you have these oxygens that are partial negative going towards the partial positive, and they do the attack and they pull it out. They surround it, right, with these oxygens that are partial negative and this positive sodium. So now you have them isolated. Okay, so all the water molecules, they're attacking, and everything's fine, and you totally dissolve the salt and you gargle and everything's great. Well, some days pass and you decide to do the same thing. However, you put a little bit too much salt and the salt falls out of solution into the bottom. So then you're like, interesting. You go and you come back later and you start again, and it still falls out of solution. And you have a way to close it so the water is not evaporating. So no water in, no water out of the system. You come back 500 years later and you stir it and it still falls out of solution. Why? The reason it will forever fall out of solution and form in the bottom of the glass is because all your water molecules are occupied. All of them are occupied. You do not have enough water molecules to perform more attacks. When all water molecules are occupied in the attack, meaning like this, they're all done, they're all attacked, they're all surrounding, and they're all used up, that is called saturation or saturation point. So they're fully saturated. The only way you are going to dissolve the remaining salt is to add more water. Never, ever, 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 ever in the entire life. You can come back 3,000 years later and it will still be down there on the bottom. So that is some of the ways that in which water interacts. Remember, this partial positive, partial negative chart is going to come into play. All right, now we go backwards to right here. Remember we did this? Now we're going to talk a little bit about macromolecules. Macro, large molecules, right? So large organic molecules. Subunits known as monomers. You can think about mono meaning one or mono. Now you can think of these as Legos, right? Just like you have Legos, you put a bunch of Legos and you link them together and you form a polymer. Polymer chains means many monomers. Here are your 4 types of macromolecules: carbohydrates, proteins, lipids, and nucleic acids. So carbohydrates, proteins, lipids, and nucleic acids. So let's take a look at carbs. When somebody says, I am on a carb diet, what does that mean? It means made up of sugars, many, 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 many sugars. So here, we're taking a look at that amazing, most important molecule, glucose. Now remember, all ATP is derived from glucose. This is the first step to make adenosine triphosphate, ATP, and ATP is the body's energy molecule. Yep. So without this, that's not good. Now, of course, you know, at least in our country, I guess due to social economic statuses and things of that nature, most people have access to glucose. If anything, typically some Americans have a little bit too much glucose. That can pose a problem, and that's why they check people's blood, known as blood glucose levels. We won't dabble into that in much detail. That's beyond the scope of this. But think of this as a Lego. This sugar has some multiple names. Of course, this specifically is called glucose, but you can call one of these a monosaccharide, one sugar. Also, they call these things simple sugars. You see that right here? Simple sugars. So a simple sugar. So a simple sugar, AKA monosaccharide, also known as monosaccharide. Put a bunch of them together and you have a complex carbohydrate, right? Simple sugar, complex carbohydrate. Now, some of these can be used for energy and other things like that. So let's take a look at proteins. Well, what are the monomers? Now, in proteins, you do not call them simple proteins. We don't call them simple proteins. That's only for the simple sugar up here. We don't call them monosaccharides because they're not sugars. Guess what? They have their own name, and there are amino acids. So we have 20 different amino acids, and amino acids are responsible for making proteins. Now, this part here is always going to be this common structure. The common structure, you may want to call it maybe the backbone of an amino acid. It's always going to be the same. That is what this color is trying to indicate. All this part here is this part here. That's why they're trying to show you that little arrow. All amino acids are going to have this carbon bonded to this double bond oxygen and the hydroxyl group. This is known as the carboxyl group. This carbon here is known as the alpha carbon, and it is bonded to a hydrogen. Then the alpha carbon is bonded to this amino group, nitrogen to hydrogens. Then this carbon is bonded to this orange thing here known as the side chain. So what is the side chain? The side chain is what really gives the identity and characteristic of an amino acid. For example, this is a side chain, 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 this is a side chain. Now, there are 3-letter codes. For example, here we have tryptophan, okay? Tryptophan is just, I'm storing an example out there. You do not have to worry about the 3-letter codes for the 20 amino acids. I will not be testing you on that because you can always just look that up. But so lucky you. I unfortunately have to know these and the RNA code for them. So that is the amino acids. Now, the side chain will dictate its characteristic. What do I mean by that? That means depending on the side chain, the amino acid may be of a characteristic of polar or non-polar. So now, if we were to get this protein and put it into water, the parts that are interacting with water would be known as polar. Then the amino acids inside that are getting away from the water would be non-polar. So here, let's take a look at lipids. Lipids are not repeating monomers. They are not. They are not like carbohydrates. They don't have a bunch of Legos, all the same Legos bonding. They're not like proteins. They're different. So let's take a look at some of the differences between them. Okay. All right, let's take a look at a triglyceride. Tri meaning 3. Glyceride for glycerol. 3 fatty acids connecting to the glycerol. Therefore, it is a triglyceride. So also known as a fat. So they have 3 of these fatty acids that are bonding to the glycerol molecule. These fats are largely known to store large amounts of energy. You can also provide like some cushioning, and also thermal insulation. Now, these fatty acids can vary in their structure. For example, they can be saturated or unsaturated. And you may have heard of that before. You're maybe into nutrition and things of that nature. You may have heard saturated fats, unsaturated fats. Well, what makes them saturated? What makes them unsaturated? If you take a look, very close look here at this carbon, it has a single bond to each of them, and it results in this nice straight line. In the case of unsaturated, you have straight line, single bond, single bond, and then what the heck? Double bond. That double bond causes a kink or bend in the chain. So why do I say this? Well, we can have certain fats in room temperature and they are stable, like solids. For example, bacon grease. You make bacon in the pan and then you leave it there, and then all of a sudden you scrape the bacon off, put it in a mason jar, and you cover it. Assuming that your kitchen is not hot, it will be a solid. Why? Because those fats are saturated and they can compact here and they can really bind to one another, making a solid. However, there are other types like olive oil. Olive oil is a liquid at room temperature, right? It's a liquid. Okay. So there are other types, for example, sterols. So a sterol, they have a 4 connected carbon ring. So I mean, they're this is what's known. Just like we saw in amino acids, the carboxyl group, the hydrogen, and the amino group, sterols have these 4 aromatic rings. So cholesterol, is a sterol. It's important. Some people always think cholesterol, bad. But the right amount of cholesterol helps make cell membranes, super important. Others are hormones. Now, let's talk about the outer part of a cell. The outer part of a cell can have, usually has, phospholipids. So now we're also going to get into the hydrophobic, hydrophilic. So we have this area right here is where we have our phosphate group. Sometimes they call it the head of the membrane. And it is hydrophilic, meaning water loving. So it's going to interact with water. It's going to be cool with water. This part here, so this is very hydrophobic. Phobia, fear, hydro, then fear of water. So we have this situation here in which phospholipids have 2 different characteristics. One is not cool with water and the other is cool with water. And we will shortly dive into that in a little more detail. The next and last one of the macromolecules, because we have covered carbohydrates, we went into your proteins, we went into your lipids, and now we're going to into the nucleic acids. In the nucleic acid, when you hear nucleic acids, you need to think of DNA, RNA. DNA standing for deoxyribonucleic acid. Ribonucleic acid. What makes them? What is the backbone of it? You have a nucleotide. So you have the sugar, the phosphate, and the base. What is the difference between DNA and RNA? Let me see if we can show you. For example, here, we have the A binds to T. And in the case of RNA, RNA does not have a T. Look at that. It has a G, an A, a C. Here we have that. We have a G, we have a C, we have an A, and we have a T. What happens is thymine gets replaced with uracil. So keep that in mind. That's an exam question. Uracil. So RNA has uracil, the U. Let's take a quick look now. With that, we've understand hydrophilic. This is going to be the polar part. Hydrophobic. This is going to be your nonpolar part. Fear of water, water loving. Okay, let's take a look at a phospholipid bilayer. The phospholipid bilayer will have the phospholipid part up here, and it will interact with water, or down here, and it will interact with something. So let's say this is a cell. This is a rough example of it. Inside the cell would be intracellular fluids. And those intracellular fluids, doesn't look like this. It doesn't look like a pond inside your cell, but they're trying to do a really good job of explaining the interactions with water or water-like substances. So water is polar, and this is water loving, and this has a polar, so polar and polar are cool with one another. They're cool. Inside, however, is your fatty acid tails. They are fear of water. They are not cool with water, and that's the reason you do not see blue in the middle of this phospholipid bilayer. You do not see that. They depict a little bit right here, this little depiction of this little shade of blue. They did a pretty good job showing that, trying to teach the student that this phospholipid bilayer is interacting with some water-like substances or water, and it is okay, but not in the inside. Now, let's take a look at this protein. Very, very interesting. So this is an integral protein, and it is in the middle of the phospholipid bilayer. What the heck is going on? Yes, we'll get into it in chapter 3, but yes, some proteins are involved to help substances go in and out of the cell that are too large to diffuse naturally. Well, if this portion is interacting with intracellular fluid, it would be safe to say that this is polar or hydrophilic. It would be safe to say that this outside part interacting with extra outside of the cell, extracellular fluid, is polar, meaning hydrophilic. It would be safe to say that inside here, in close proximity to the fatty acid tails, in which they are hydrophobic, that this part of the protein is hydrophobic. And that is what I was trying to talk about when we looked at the protein. It is the side chain characteristic that will either be out here or in here. That's all dependent on that R group, right? So that is why I jumped into the water, talked about water, then talked about water characteristics and all these other things, so we can come back here and get a good grasp of this whole concept. We already covered this, just in case. We already got this. We already covered 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 this. And I don't know really what to say too much about this part here. Water on Mars, okay? Yeah, so I guess there's some evidence that shows, I mean, I'm not really knowledgeable on this stuff, but maybe there was water. We talked about earlier in your part 1, so please take a look at part 1. It starts out with Mars again. So it says liquid is not present on the surface of Mars. Okay, yeah, we know that. Clues to ancient water covering Mars can be seen all over on the surface of the planet. So yeah, there are some things that we discussed earlier that do show that there must have been some sort of a liquid possible, like water, that made certain things that are very similar to rivers and streams that are on Earth. But as far as us knowing it was water, we hypothesize, right? That we make an educated guess until we have more evidence to say it was definitely water. All right, let us talk about pH. So here they say pH is the concentration of hydrogen ions. Remember, ions, charge. Remember that sodium was positively charged? We'll look at this hydrogen, positively charged in solution. So concentration of hydrogen in solution. What is concentration? The amount of stuff per volume. So the more of this in a certain volume, the stronger the acid. We'll get into that. So let's zoom in and let's start breaking it down. So the pH scale goes from 0 to 14. 0 to 14, okay? In the middle of that is 7. That is neutral, for example, pure water, right? Not water from the tap, Not your alkaline water, not that. Pure water, meaning like deionized, removed of ions, deionized water. You'll see it on the shelf, deionized. So that would be 7, should be 7. And then there's other stuff in lab grade, nano water and all these other things, but we won't get into that. So 0 to 14. Now, I'm going to say something and it's going to sound a little weird because of the way we are taught numbers. Normally, we are always taught that from 0 as, you gain more as you are going up this number line, right? Well, that is not the case in terms of acid. In terms of acid gets stronger as the value approaches 0. Yes, that is correct. Acid gets stronger as the value approaches 0. For example, battery acid. Please make sure you do a little research and know how to properly charge a battery or give someone a proper jump. On the rare occasion, they do sometimes explode. Okay. Another one would be hydrochloric acid. That is down here, 0. Very dangerous to the human body. The same can be said in terms of danger to the body here. Way up here, it's 14, and this is considered a very, very strong base, like liquid drain cleaner. All right, if you're going to use this, be careful not to get in your hands. It's best if you wear some gloves. So this can be dangerous. That's how come it's able to dissolve all that nasty hair down in there in your old G.I. Joes, in your toilet or shower, wherever they go. You have your liquid drain cleaner. Okay, let's go back. I really, really like this because I think they do a great job displaying what's going on in terms of pH. So let's take a look at the strong acid. There are a bunch of these ions. Ions are charged. Specifically, we're looking at hydrogen ions. So the more H's you have, the stronger the acid. Okay, let's take a look at orange juice. Oh, we have some good amount of H's. Oh, look at that. We have an OH known as a hydroxyl. As we move over here to coffee, coffee. 1, 2, 3, 4 hydrogens, 2 hydroxyls. Oh, we still have more hydrogens. So yes, we are still in acid. And we're looking at 2 OHs. Why is coffee an acid? Because caffeine, the correct term for caffeine is called caffeic acid. So coffee is an acid. That's why it's not good to really be drinking coffee on an empty stomach. So we have pH 7. Now let's take a look at this. Now, please do not think that this is just a representation. Do not think that coffee only has 1, 2, 3, 4 hydrogen ions and only 4 hydroxyls. That is not the case. What they're trying to show you is that, hey, look at this introduction of hydroxyl ions, and we keep approaching, we're moving up towards 7. Now let's count here. We have 1, 2, 3, 4 hydrogen ions, 1, 2, 3, 4 hydroxyl ions. 4 and 4, perfectly neutral, right? Okay. Now, as we move up to baking soda, 1, 2, 3, 4 hydroxyls, 2 OHs. And we move up over here and we see all the OHs there, OHs, hydroxyls. There are not any H pluses. Very interesting. It is the exact opposite of, this is the exact opposite of this. No OHs. No H's. Very strong, very dangerous. Now, please, ladies and gentlemen, do not mix things that are acids with chlorines. Do not do that. Do not mix things that are acids, things that are cleaners. They don't have any cleaners here. Do not mix things that are acids with things that contain chlorine or very strong products, cleaning products, because you can then have chlorine gas, and that can be very toxic and that can be deadly. So please don't do that because you can die. There are many things that can kill you in your home if you do the wrong thing. All right, Now let's talk about this. Where do these come from? Okay, water briefly splits into OHs and, I'm sorry, hydrogen ions and OHs. Now these, they're not only coming from water, sometimes they can, but usually they're coming from other molecules, okay? They're showing you that water can briefly split. Last slide here. Since we were talking earlier on what constitutes life, those 5 properties, let's briefly talk about a virus. Now, the reason people have the name a computer virus is because viruses were the first things on Earth to hack. What do I mean by hacking? Well, most viruses on Earth cannot replicate. And at the beginning of this presentation, meaning part 1, one of the properties of life on Earth is being to replicate. Flowers, ants, whatever. They must be able to reproduce. Well, since viruses are unable to reproduce, they have been classified as non-living entities. I will say, however, there are some viruses that have been discovered that can self-replicate, making them considered to be alive. So how does something that is not living replicate? A virus will enter your cell, and then the cell it doesn't recognize, your immune system kind of doesn't recognize it yet. It goes in what is known as the replicative machinery. Think of a factory in your cell that's responsible for DNA and RNA replication, and it goes in that factory and it slips in its code, and then before you know it, you are making the virus. Stupid cells. Yes, that is correct, and we end up killing ourselves. It's true. We end up making the virus. So then it's too late, and then we figure out with a fever that, oh gosh, I have the flu. Of course, in the case of COVID-19, since that's a very unique virus, there are all different kinds of symptoms. Some people never get fevers. So that's the situation on viruses. In the case of prion disease, prions are infectious proteins disease, or a misfolding of proteins. Now, these are very, very, very dangerous. For example, mad cow disease, that was a prion disease. Or in the case of humans, it's called Creutzfeldt-Jacobs disease. And that causes people to experience damage to the central nervous system. And there is no cure, and usually results in death. Yes. So that is the end of our presentation. Ladies and gentlemen, do we have any questions? Perfect timing, ended at 7:02. Couldn't have done it any better. Questions? All right, well, I will stop it here so I can save on data.